This lab is about the purification of S3 and pGLO plasmids. The materials consist of a pipette pump, serological pipette, culture tubes, inoculation loops, waste container, albient broth, and agar plates from S3 plasmids and pGLO plasmids, vortexer, microcentrifuge, micropipettes and tips, microcentrifuge tubes, resuspension solution, elution solution, lysis solution, neutralization solution, wash solution, and green mini columns. For part 1, label 2 culture tubes with your initials and date. Label the tubes as follows: S3 mini and pGLO mini. Then, use an aseptic technique to transfer 4 milliliters of LB ant broth into each tube. Using a sterile loop, scrape 1 colony transformed with the S3 plasmid from the surface of the LB amp positive plus 3 plate. Transfer the loop into the culture tube labeled S3 mini. Swirl the loop around to release the bacteria and recap the tube. Repeat this for the LB amp plus pGLO into the pGLO mini culture tube. Recap that tube as well. After that's complete, place the culture tubes in a shaking incubator overnight. For part 2, label capless collection tubes S3 mini and pGLO mini, as well as 2 green columns with the same thing. Place each green column in its corresponding collection tube. Transfer 1.5 milliliters of each culture into the corresponding 1.5 milliliter capped microcentrifuge tube by pipetting 750 microliters twice. Close the tubes. Place the tubes in the microcentrifuge in a balanced pattern. Put the hinges of the tubes facing outwards so the pellet will be easy to locate after centrifugation. Centrifuge the tubes and the cultures at 1 minute at top speed to pellet the bacteria. Locate the bacterial pellet and remove the liquid or the supernatant from each tube using a 100 to 1000 microliter micropipette. Add another 1.5 milliliters of each bacterial culture to the corresponding 1.5 milliliter capped microcentrifuge tube that already contains the bacterial pellet. Spin the tubes again for a minute and discard the supernatant once again. Now, using a fresh pipette tip each time, Pipette 250 microliters of resuspension solution into each tube. Resuspend the bacterial pellet by pipetting up and down, or by vortexing. Next, pipette 250 microliters of lysis solution into each tube and mix by gently inverting 6 to 8 times. Do not pipette or vortex. Pipette 350 microliters of neutralization solution into each tube and mix by gently inverting 6 to 8 times. A precipitate should form. Spin the tubes in the microcentrifuge for 5 minutes at top speed. Pipette the supernatant from the centrifuge tubes into the corresponding green mini column. Spin the columns in the capless collection tubes in the microcentrifuge for 1 minute at top speed. Take the column out of the capless tube and discard the liquid from the collection tube. Replace the column in the collection tube. Pipette 750 microliters of wash solution into each column. Spin the columns for another minute, discard the flow through, and go again for 1 minute. Label one microcentrifuge tube S3 plasmid and another pGLO plasmid. Transfer each column to the corresponding clean 1.5 milliliter capped microcentrifuge tube and discard the collection tubes. Now, pipette 100 microliters of elution solution directly onto the column bed of each column. Allow the solution to absorb into the column for 1 to 2 minutes before spinning the columns in the microcentrifuge for 2 minutes at top speed. Measure the volume of the solution eluted from the column using a 20 to 200 microliter adjustable volume micropipette. If the lab was done correctly, both liquids should now contain the plasmid, but without moving on to lab 5.4, it is hard to determine if half the lab was done correctly. For the pGLO plasmid, it should glow under UV light, but for the S3 plasmid, you'll have to wait and see. Some things to avoid are reading the protocol incorrectly and missing a step and adding a solution, forgetting to let the elution solution absorb into the column near the end of the lab, and try to avoid accidentally pipetting the pellet when the protocol calls for just pipetting out the supernatant. Just leave a little liquid at the bottom of the tube with the pellet. Finally, make sure you balance the microcentrifuge so the machine does not break. Thank you!